Hi, I'm Larry Johnson. I am a professor here at Texas A&M University, and today we're going to talk about the liver, the gallbladder, salivary glands, and pancreas. We'll be talking about the glycogen storage and use, sugar and proteins, liver zonation, bile duct, bile canaliculus, hepatic duct, gallbladder, epithelium of the GI tract in review.
The hepatocytes here, there's this nucleus and then there's cytoplasm there, is shown as glycogen. Stained for glycogen. You can see the kind of red structure is glycogen. This we can see is glycogen 2. It's not stained red, it's just a black and white image. But this is 2 hours after a fast. And you see 8% of the hepatocyte is glycogen. However, after a 24-hour fast, then the glycogen has been depleted mostly. And so this illustrates that the liver plays a role in the blood sugar concentration on a daily basis. And it makes up for if you don't have sugar intake from the digestive tract, it can make up that temporarily for during the day's time. Sugar and protein, here we have sugar comes up, glucose comes in, stored as glycogen, and then is broken down, degraded to glucose again. And then also you got amino acids coming in. They make proteins, and then the proteins could be albumin, fibrinogen, prothrombin, lipoproteins that are produced there. And this is in the endocrine aspect of it, and that's separate from the exocrine aspect of it, where you have bile is being produced in the bile canaliculus. And here you can see where the two surfaces are separate. You have the sinuses, which are the endothelial cells and the Kupffer cells, lining them, and then you have the bile canaliculus, the yellow that you're seeing that then gives rise to a bile duct. Now, the liver has different zonations. The classical lobule is the one I mentioned before that we see. There's also a portal lobule with a triad in the center, because the triad is a source of blood that goes to the central veins, and one triad could go to multiple lobes. Another way to look at it is the ascens, and that is the different layers. The first layer, if this is a triad and triad here, both of these can contribute to the central veins in both of these. And so you have the zone, the first zone, second zone, and third zone. So there's the classical lobule, portal lobule, and the ascens. And the ascens is the main one that's important. If you have liver damage due to a toxin, it will kill the ones in zone 1 first. And it would remove the toxin from it, so these cells are spared. However, if you have some kind of toxicant that will use up oxygen, these cells in zone 3 are already deprived of oxygen, so the toxin would kill these cells first. So if you have some animals that have died at a feedlot, and you look at the liver, and you'll be able to tell which cells are destroyed first, then it would tell you what kind of toxin you might be dealing with to help save the other animals in the herd. Here we see the liver. There's a central vein. You can see the kind of pink here are the hepatocytes. The solid kind of reddish color are the red blood cells that are in the sinuses. And here we can see many Kupffer cells throughout that has a role of cleaning the blood in the liver. Here we see another one. This is a gallbladder right in through there, but there's a liver here, a nice liver that we can see the triad, the 3 different components. You have the hepatic vein, hepatic artery, as well as the bile. And that's the triad components of it. And remember, the blood is going to the central vein, and the bile is coming back toward the triad. And here we can see the different cells there. We got the Kupffer cells, which cross over through there. Got endothelial cells, as well as hepatocytes. In the bile canaliculus, you can see the occluding junctions, the zonal occludens and zonal adherents. which are preventing things from going between adjacent cells in the bile canaliculus. Here we can see the bile canaliculus right in through there. We can see it in through there, and through there. We can see the bile canaliculus between adjacent cells, and that's a different surface from that of the blood luminal surface that we see here and here, and also we can see some bile ducts as well. Now, if you stain a piece of liver for proteins that are in the bile canaliculus, this is what you see, that indeed the bile canaliculus touch all the hepatocytes and can be seen throughout. However, if you look at the bile duct is where you have concentration of the bile, but you don't have it distributed throughout like you do the bile canaliculus. The bile canaliculus has to touch each cell if the cell is going to contribute to the bile. Now, in the bile canaliculus, you have different things that are there. You got cholesterol in there, epidermal growth factor, insulin, and IgA. Body fluids have antibodies in it to help in the immune response. Also have bile salts and bilirubin that's located there too. So you have the bile canaliculus giving rise to the bile duct. There's a duct on the bile duct, as you can see. So it giving rise from here to the bile duct, which has cube cells in through there. Then the bile goes through the common bile duct, and then it goes into the hepatic duct, you might say, a common bile duct, and then it goes through the cystic duct into the gallbladder itself, where it's stored. And here we can see the cystic duct, the hepatic bile duct, and this is the portal vein that is bringing the blood to there. And so here we can see in the common bile duct, you can see simple columnar epithelium, the same as what you see in the cystic duct, and also it's the same as what you see in the gallbladder itself. Now, in the cystic duct itself, you can see it's kind of closed there because you kind of have a swirling of the lining, the connective tissue, and the epithelial lining, as well as smooth muscle in through there, and that kind of prevents the collapse of the duct whenever you're changing the different pressures that might be involved there. So this is the cystic duct, which is the duct that goes to the gallbladder, and it has a little valve in there that regulates the flow of that. Here we see the gallbladder, some smooth muscle, a lot of connective tissue, and simple columnar epithelium covering the lamina propria. We can see the real specimen. This is liver out here, but then you can see a simple columnar epithelium. In the lamina propria, there's lots of plasma cells associated with producing antibodies that would go into the bile that we mentioned before. You see how the enfoldings in through there increase the surface area, and also note that the cells can be separated out because actually it is pumping out water out of these. So it's pumping out electrolytes and pulling the water with them to concentrate the bile. Here we can see a higher magma, one of those. You can see the simple columnar epithelium that's there. We see a thick connective tissue layer. We also see a serous cavity with mesothelial cells on the surface that we can see right here and here, a connective tissue below, and more connective tissue when it has some smooth muscle in there as well. Now, one of the things you need to do with the GI tract is to be able to identify different cells on the surface. And here's the cardiac region. These are the three regions of the stomach. This is the fundic region where you have surface mucus cells on the surface. You have protocells, chief cells in through there. Pyloric region, this is PAS staining. the intestine, simple columnar epithelium with goblet cells, and then here you can see the gallbladder, again, simple columnar epithelial cells to be able to see the different ones. The mucosa that we want to look at is the surface epithelium, lamina propria, as well as the muscularis mucosa that's down below.
We want to acknowledge the original source of various micrographs, images, and drawings that might have been used in these presentations, and that original source have been identified with the figure that was used. This is the end of the liver, gallbladder, salivary glands, and pancreas with the glycogen storage, use, sugar and proteins, liver zonation, bile duct, liver canaliculus, bile canaliculus, hepatic duct, and the GI epithelium in review.